السلام عليكم أعزائي الطلبة. اليوم نتكلم عن موضوع الإميونيزيشن. تعرفون الحدث خلال العام 2020 هو appearance of COVID-19، والعالم حاليا كله يحاول إيجاد سيطرة لهذا الفيروس. حاليا بعض اللقاحات تم تصنيعها من قبل عدة شركات. الغاية من اللقاحات هو to control COVID-19 infection. بعض الدول تم استخدام هذا اللقاح وبنجاح، والآن انتظر دخول اللقاح إلى العراق إن شاء الله عن قريب. So the teaching objective is to know the distinction between passive and active immunization and their examples, to distinguish between artificial and natural means of immunization, to know the application and the problems of artificial passive immunization, and to know the application and problems of artificial active immunization, and to know the modern approach of immunization. So what is the immunization? ما هو التطعيم؟ Immunization is the mean of providing a specific protection against most common and damaging pathogen. The mechanism of immunity depends on the site of pathogen and also the mechanism of its pathogenesis, how the microorganism will enter to the body and induce disease. So, if the mechanism of pathogenesis involves endotoxins that produced by gram-positive bacteria, the only mechanism of effective against it would be the neutralizing antibodies that would prevent its binding to the appropriate receptor and promoting its clearance and degradation by phagocytes. Alternatively, if the pathogen produces disease by other means, the antibody will have to react with the organism and eliminate it by complement-mediated lysis or phagocytosis and intracellular killing. However, if the organism is localized intracellularly, it will not be accessible to the antibodies. Antibody ما يقدر يدخل للسيلز. While it remains inside, and the cell harboring it will have to be destroyed, and only then the antibody have to exert their effect. Most viral infection and intracellular bacteria and protozoa are examples of such pathogens. In these cases, the harboring cells can be destroyed by element of the cell-mediated immunity, or if they cause the infected cells to express unique antigens recognizable by antibody, the antibody-dependent and complement-mediated killing can expose the organism and eliminate the bihumoral immunity. Alternatively, the cells harboring intracellular pathogen themselves can be activated to kill the organism. Such is the case of with pathogens that have the capability of surviving within the phagocytic cells. أكو تعاون بين cell-mediated immunity and humoral immunity. فبعض الأحيان تكون intracellular organism should be expressed on the surface of the RBCs cells. So, expression of the antigen on the surface, it will be recognized by antibodies and will mediate killings. So, the immunity could be a natural resistance or acquired. Specific immunity can result from either passive or active immunization, and both modes of immunization can occur by either natural or active artificial processes. Now, what is the types of immunity? First, passive immunity. Immunity can be gained without the immune system being challenged with an antigen. by transfer of serum or gamma globulin from the immune donor to non-immune individuals. Alternatively, immune cells from immunized individuals may be used to transfer immunity. Passive immunity may be acquired or naturally or artificially. So, passive immunity could be humoral immunity or cellular immunity. And examples of these are natural acquired passive immunity. The immunity is transferred from mother to the fetus through placental transfer of immunoglobulin G or colostral transfer of IgA from milk. Artificially acquired passive immunity is the immunity is often artificially transferred by injection of gamma globulin from another individual or from an immune animals. Passive transfer of immunity with immunoglobulin or gamma globulin is a practical in numerous acute infections like diphtheria, tetanus, measles, rabies, or could be for poisoning by insect, reptile, bite, botulism, as a case of prophylactic measure in case of hypogammaglobulinemia. In these situations, gamma globulin of human origin is preferable through specific antibodies arise in another species. Examples of another realm like horse. The effective and used in some cases. While this form of immunization has the advantage of providing an immediate protection, it is effective for a short duration only, فترة قصيرة, and often result in a pathological complication, such as serum sickness, which is characterized by rash, fever, arthralgia, vasculitis, nephritis, etc. Or anaphylaxis. Homologous immunoglobulin may carry a risk of transmitting certain diseases like hepatitis, HIV, and so on, and other blood-borne disease. Passive transfer of cell-mediated immunity, the immunity is transferred by cells and not by antibodies, can also be accomplished in certain diseases like cancer and immunodeficiency. However, it is difficult to find histocompatible matched donor, and there is a severe risk of graft versus host disease. The other type of immunity is the active immunity. This is referred to immunity produced by the body following exposure to antigen. Could be a natural acquired active immunity or artificial acquired active immunity. For the natural acquired, exposure to different pathogens lead to subclinical or clinical infection, which is normally result in a protective immunity response against these pathogens. Best example here, COVID-19, where about 80% of our population could be infected without any clinical signs or mild clinical signs, and these provide some sort of immunity. While for artificially acquired active immunity, immunization may be achieved by administration of live or dead pathogen or their component. Vaccine used for active immunization consists of a live attenuated vaccine which is capable of producing very mild or no symptom at all. Killed whole organism, microbial component, or secreted or detoxified toxins. All these are different types of vaccines available. Live vaccines. Some vaccine can be safely given alive for not so dangerous pathogens. Live organisms are used for immunization against a number of viral infections. كنا نعرف اللقاحات اللي تعطى في فترة الطفولة. صراحة الأغلبية لهذه اللقاحات هي live attenuated viruses. For example, live vaccine for measles, mumps, rubella, and newly designed vaccine for chickenpox are used routinely. A live bacterial vaccine consists of a strain of Mycobacterium bovis, Bacillus calmette, and gaurine, is used against tuberculosis in some African, European, and Asian countries, but not in many others. داخل العراق نستخدم البي سي جي اللي هي Bacillus of calmette and gaurine, whereas many studies have shown that the efficacy of BCG vaccine, a number of studies also cast doubt on its benefit. Actually, the best benefit for BCG vaccine about 30% protection. Usually because this type of vaccine is of bovine vaccine. So what is the benefit of live vaccine? Usually the live attenuated vaccine give longer duration protection. Usually produce a self-limiting non-clinical infection and leads to subsequent immunity, both humoral and cellular-mediated immunity. The latter being essential for intracellular pathogen, usually the viral pathogen. However, they carry a serious risk of causing overt disease in immune-compromised individuals. لذلك احنا قبل ما نعطي اللقاح للأشخاص نسألهم إذا عندهم any disease associated condition, especially for immune system deficiency. Furthermore, since live vaccines are often attenuated by passage in animal or thermal mutation, they can revert to their pathogenic form and cause serious illness. يتحول إلى natural one by mutation, wild one. It could be a same variant or even more virulent vaccine. It is for these reasons polio live vaccine, which was used in many years ago, has been replaced in many countries by the inactivated vaccine, Salk vaccine. حاليا داخل العراق is the inactivated vaccine. Another type of vaccine was the killed vaccine. These consist of a whole organism inactivated or killed by heating process, addition of some chemicals, or by UV irradiation treatment. Many killed viral and bacterial vaccines are available. Some of these are used to immunize people at risk. Examples, the routine seasonal influenza or for hepatitis A viruses, while others are used to immunize travelers to different countries and the suffer, like cholera vaccine and typhoid vaccine. Pertussis, which is named whooping cough, is a whole bacterial vaccine, was used routinely until a few years ago, but it is due to serious side effect, it has been replaced by the formulation of a cellular component. Another type of vaccine is called the subunit vaccine. Some vaccines consist of a subcomponent of the pathogenic organism, usually proteins or polysaccharides. Since polysaccharides are relatively weak T-independent antigen, and produce only IgM responses without immunogenic memory, they are made more immunogenic and T-dependent by conjugations with the proteins. So lipopolysaccharide will be formed. Examples of these vaccines are Haemophilus, meningococcus, and pneumococcus. This we call triple vaccines used for prevention of meningococcemia. Another example is hepatitis B, rabies. Vaccines consist of antigenic proteins cloned into a suitable vector. Could be an yeast or E. coli. These subunit vaccines are designed to reduce the problem of toxicity and risk of infections. Hepatitis B surface antigen اللي موجود حاليا ويستخدم regularly highly effective vaccine is a type of subunit vaccines. So, when the pathogenic mechanism of an agent involves a toxin, a modified form of a toxins we call a toxoid is used as a vaccines. Examples for diphtheria and tetanus. Toxoids, although lose their toxicity, but they remain immunogenic. A new approach for vaccine production, which is called the other novel vaccine. A number of a novel approach to active immunization are in the investigative stage and are used only experimentally. These may include anti-idiotype antibodies, and we talk about how to produce an anti-idiotype antibodies. A DNA vaccine, the best example is the messenger RNA vaccine produced by DNA technology for COVID vaccine, and immunodominant peptides recognized by MHC molecules and may be available in near futures. What about anti-idiotype antibodies against polysaccharides antibodies, which produce a long-lasting immune response with an immunogenic memory? Viral peptide genes cloned into vectors, when injected, transfect host cells and subsequently produce a response similar to that produced by active live attenuated vaccines. So both cellular and human immunity produced by these type of vaccines. Immunodominant peptides are simple and easy to prepare, and when incorporated into MHC polymers, they can provide both humoral and cellular responses. So by these different types of mechanism, we can obtain a potent vaccine which evoke both a humoral and cellular immune response against infection. A newer adjuvant formulations include synthetic polymers and oligonucleotides. Most adjuvants recognize toll-like receptors and thus activating mononuclear phagocytic cells and inducing selective cytokine production that can enhance T helper 1 or T helper 2 immune responses, depend on the nature of the adjuvant itself. This table shows the selected adjuvant in a clinical or experimental use. Here we see different types of adjuvant and where the experiment only used. Salt adjuvant like aluminum hydroxide, aluminum phosphate, they cause slow release of antigen. Synthetic particles like liposomes and polylactates, slow release of antigens, polynucleotide, also bacterial product and cytokines. The main role is the cytokines, the activation and differentiation of T and B cells and the antigen presenting cells. The protective immunity conferred by a vaccine may be lifelong, like vaccination by MMR vaccine, measles, mumps, rubella vaccine, or smallpox vaccine. These are life attenuated vaccine which give a very long or lifelong immunity. Also used for tuberculosis and yellow fever, or may last as little as a few months, like cholera vaccine. These are killed vaccine. The primary immunization may be given at the age of 2 to 3 months, like diphtheria, pertussis, tetanus, polio, or 13 to 15 months, like mumps, measles, and rubella. The Iraqi schedule for immunization was a little bit different. They give BCG vaccine within the first week, also give subunit vaccine for hepatitis B virus within the first week, followed by a schedule of 2 months, 4 months, and 6 months for DPT vaccines, followed by a 9th month for mumps vaccine, and the triple vaccine at the 12 or 13 month, which is the MMR vaccine. A booster dose of vaccination was recommended regularly for polio, For the females to prevent congenital rubella infection, the vaccines is given in the secondary school years at about 12 to 13 years, and this is for prevention of development of congenital rubella infection. This is the schedule of Iraq. The currently recommended schedule for routine immunization in the USA, which is recommended by CDC, Center for Disease Control, is summarized in a figure shown in the PDF provided with the lecture. Now, when we give a prophylactic vaccine and when we give a therapeutic vaccine. Usually, most vaccines are given prophylactically. I mean, prior to exposure to pathogens to prevent entrance of the virulent infection. However, some vaccine can be administered therapeutically. I mean, post-exposure to the virulent invading agent like rabies virus. The effectiveness of this mode of immunization depends on the rate of replication of the pathogen, inoculation period, and pathogenic mechanisms. For these reasons, only a booster shot with tetanus is sufficient if the exposure to the pathogen is within less than 10 years. And if the exposure is a minimal, like wounds are relatively superficial. In a situation where a pathogen has a short incubation period, the pathogenic mechanism is such that only a small amount of pathogenic molecule could be a fatal, like tetanus and diphtheria infection, and or a bolus of infection is relatively large. Both passive and active post-exposure immunization are essential. Passive prophylactic immunization is also normal in cases of defect in the immune system such as hypogammaglobulinemia. بالحالة احنا ما نقدر نعطي live attenuated vaccine because there will be a reactivation of the attenuated and may cause severe disease process. Now, what is the adverse effect of immunization, which could depend on the type of the vaccine used? Usually, active immunization may cause fever, malaise, and some discomfort at local injection site. Some vaccine may cause joint pain or arthritis, like example, rubella vaccine. Convulsion may occur, which it could be fatal in cases of pertussis vaccine, or neurological disorders associated with influenza vaccines. Sometimes with the product of the cells that inoculated by the vaccines, like an egg preparation of some vaccines may cause allergy. So allergy to eggs may develop as a consequence of viral vaccine produced in eggs, like for measles, mumps, influenza, yellow fever vaccines. Sometimes booster shot may result in a more pronounced inflammatory effect than the primary immunization. The noticeable and serious side effects documented have been those following the DPT vaccine. Most of these are attributed to the whole pertussis component of the vaccine and have been eliminated since the use of a cellular pertussis preparation. So it depends on the type of vaccine, whether it's live attenuated or killed vaccine, and whether the preparation of the vaccine associated with certain allergenic substance like an egg preparation of some vaccines. All these may cause an allergic reaction. Sometimes a virulent vaccine may cause a permanent or temporary brain damage due to the neurotropic effect. So these are some side effects of these vaccines. This table shows the approximate rate of adverse event occurring within 48 hours of diphtheria, tetanus, pertussis vaccination. So there is a local redness, swelling, and pain. mild moderate systemic effect like fever, drowsiness, fretfulness, vomiting, anorexia. More serious systemic effect like persistent crying, fever, collapse, convulsion, acute encephalopathy, and permanent neurological defects. And these the frequency noted following by the appearance of the adverse events. Thank you for listening.